develop and promote new collaborations, new areas of investigation, maybe highlight the work of junior investigators as much as we can as well, many of whom you've read their proposals and now they're actually conducting the research. So thank you. And then obviously if you have ideas, things we can incorporate in these conversations, we're happy to do that. But we just wanted a place for us to get together and talk science related to clinical and population sciences. But we anticipate in time that there will be cross-cutting discussions with the other cores as well, especially related to cure and other things. So today we wanted to start off by, I'll introduce you to Larry, who I don't know if all of you have met Larry Jean-Pierre, who is Warren Jean-Pierre, who is our new project director for our STAR cohort, which is the young women with HIV cohort that's RO1 funded, 5 sites in the southern US. So we are one of the sites, and it's kind of building on what we've done with WISE. But she's the project director for STAR. And so she's also helping us as our manager for our CPS course. So communications for us, please feel free to direct it through her. She's a PhD in sociology and she's a qualitative researcher. So we're very excited to have her with us. And today, Manya and Amanda will be talking to us about 2 of the cohorts that we're fortunate to have here in the district, the DC cohort that Amanda is leading, and then our new clinical trials unit that's building on other clinical trials units that we've had in the district. And then this is really by way of sharing information about what's available, a little bit of background about what's currently being done, and then possible areas of investigation that people might think about based on what they hear today. I think we're recording, and I know that from our earlier Women and HIV Sick, there are a lot of people who are very interested in hearing about the data variables that are collected and thinking of questions we can ask using the data that you guys are collecting or will be collecting in the future. So I'll hand over to, I think, I'm not sure who was going first. Manya, was it you?
I think why don't we go straight to Amanda,
Okay,
and then
Amanda,
we'll wrap back around. Thank you so much.
Okay, thank you.
Thanks. I'm just going to share some slides, which I'm looking at the audience, and many of you guys already know about the cohort. So, oh, that's showing you the wrong thing, isn't it? Let's see. Are you seeing slides?
Yep. Yep, we do.
Okay. All right, there we go. Sorry, I have 2 screens. I just want to briefly kind of, this is really 10,000 foot level, talk about the cohort and hopefully have time for any questions that arise after presenting. I see Anne is also on. Anne Monroe is the project coordinator for the DC cohort and works very closely with me. So in general, what is the DC cohort? A lot of people ask that. So basically, we have established a database of individuals who are living with HIV and consent for us to receive information from their outpatient electronic health record at 15 clinical sites in DC. And I'll show you what those sites are in a few minutes. But that essentially includes all of their kind of baseline risk factors, diagnoses that they receive at that clinic, whether it's HIV related or not, all lab results that they receive at that clinic, treatments, and selected procedures. And once individuals are consented to be in the study, we do have a kind of a network, a team of research assistants at the sites who do baseline data extraction, but most of the data then comes to us on a monthly basis through automated electronic health record extractions into a centralized database that is now managed by our GW Biostatistics Center. So this is a citywide initiative. It includes a wide range of clinics, including academic clinics, community-based clinics, including FQHCs, as well as the VA, which is our one government clinic. We really are excited. We now are 10 years into the study, and we have one of the largest HIV cohorts to date, as well as one of the only urban cohorts that we're aware of. So the other really important aspect of the study is that it's conducted in close collaboration with the DC Department of Health, with the HOSTA, the HIV/AIDS program, where we do periodic linkages of our electronic health record data with their surveillance databases. So just to give you a sense of where we are in terms of enrollment, we have about 14,000 or so patients that are cared for across the 15 clinical sites. So you can see that a large proportion of them have already been approached to be in the study. We have over 11,000 participants who've consented. We have longitudinal data on all participants from those 15 sites, which equates to approximately 59,000 person years of data. So we really are excited that we have such a robust data set that's available to answer really myriad questions about people with HIV. We also, so the kind of the inception of the study was that it was an observational cohort, but over the time we have kind of built out various sub-studies. So the first is the molecular epi sub-study that was actually just completed in November of 2020, and actually Sebla was also a part of that study. We recruited over 200 individuals who were either chronically infected and viremic or newly diagnosed with HIV who were within the cohort. And the goal there was to try to characterize their phylogenetics, to look at transmission patterns and behavioral risks for onward transmission. We also have a study that Anne is leading on PROS, which is patient-reported outcomes. These are going to be data that are collected through tablets that will really complement the electronic health record abstraction data. And right now, the modules in the PROS are focusing on mental health and substance use. And we hope to start collecting data probably in the next week or so from all participants, and then they will be asked to complete this survey annually thereafter. We are also launching RO1. That's a collaboration with investigators at UVA, where we're doing a cluster randomized trial of an mHealth app to look at its use in improving retention and care and viral suppression. We'll be enrolling 560 people across the various sites to do that study. And then lastly, during the pandemic, we launched a COVID-19 cross-sectional survey where we're collecting data via REDCap, and we then can assess the incidence and impact of COVID among people living with HIV. Our goal right now is to collect data on about 1,000 people. And we have a telehealth component to that, which has been driven by one of the investigators at Georgetown. And with all of these studies, we also, the idea is that we collect these additional data and then we link them to that kind of parent database that we're capturing on everyone. We were funded about 6 months ago for our next iteration of the cohort, and we will be doing a lot more data linkages. So in addition to having the data from the sites, we also will be collecting, continuing our linkage with HOSTA, but we also are going to be able to access Medicaid claims data, pharmacy data, cancer registry data, and data from CRISP, which is our regional health information exchange. So again, we really are hoping that we're building as robust a platform as we can to be able to answer many, many questions about our patient population here in DC. This is just showing you the enrollment by site. You guys can look at this, and I can send out the slides if people want to get more detailed and granular about how many patients are at a particular site. What we've done in the past with individuals who are interested in using our data is that if you provide your eligibility criteria for the question that you want to answer, we then will query the database and get back to you as to the feasibility of answering that question, as well as hopefully guide you to particular sites that may have more patients that fit that criteria, so that you can work directly with the sites if you want to do some additional data collection. So as far as data availability goes, we have kind of two different levels, well, actually really three. We have site level data. So all of the sites obviously own their own data. We provide them every 6 months with kind of an aggregate look at their data. And then we really are encouraging through the CFAR these cohort-wide data requests. So if you're looking at using data from the cohort at more than one site and you're external to the cohort, then we ask that you identify somebody within the either a site PI or a member of our Data and Statistics Coordinating Center like myself or Anne or Alan Greenberg to work with you. We do require that the executive committee, which is made up of all the site PIs and the DSCC, review your concept sheet and approve it. We also allow all site PIs to have an opportunity to be co-authors on particular concept sheets. And then we work with you all to make sure that you get the data that you need and that we have data use agreements and data management plans in place. So this is a rather dated schematic of how our cohort-wide analyses work. And I actually think I'll just skip this for the sake of time, but if people have specific questions, I can go through this. So in general, we do have a data dictionary that's available upon request. But what we do have more easily available on our DC cohort website is a data request form, the concept sheets, and then we work with you also to develop those data use agreements. We have a small but mighty analytic team that can help with analyses. We have access to doctoral level biostatisticians who are also able to work with investigators. And again, just to reiterate that all cohort-wide concepts have to be reviewed and approved and prioritized by our executive committee. So in many of the CFAR Pilot Award announcements, you will see reference to well, I guess I should have changed this. It's no longer the Max and the Ys, but also to the DC cohort. And so we do encourage collaborations. You know, if you want to do a secondary data analysis, please feel free to come to myself or Anne. And then again, we will support you analytically to make sure that you are able to conduct your analysis as planned. So I think this is my last slide. Again, just kind of highlighting the unique DC cohort features. So we do have this really kind of automated electronic data capture. This is a population-based longitudinal sample. I know I went through it quickly, but the sites are inclusive of pediatric sites to really older individuals. We have a diversity of types of sites that are participating. We have this unique DC cohort, DC Department of Health database linkage. We also have a clinical dashboard, which I haven't shown you today, that we will be looking at the use of that. And again, just to reiterate that this really is meant to be a public health and research resource, not only for the investigators in the cohort, but for those in the CFAR and in the DC area. So with that, I think I will stop, and there's the website in case anybody wants to check it out, and that's my email if you have any specific questions. And I can send these, I guess, should I send them to Laurie maybe to distribute?
Yes, thank you. That would be great. And then it's Larry.
Oh, Larry, sorry.
Thank you so much, Amanda. I think I want to just highlight how much I appreciate your going over this. I'm fortunate to be able to work with you. I know a lot about the DC cohort, but every time I see it, I notice there's more and more capacity and it has more and more richness of potential. And so everybody on this call is really high level people who you may not be able to work with a cohort, but you might have students or junior faculty who might have interesting ideas. And just reminding us of this amazing thing that is a local resource is just wonderful. So I'm always excited to hear what you've done. I really appreciate it. I don't know if there are questions so that Amanda can then relax a little and have her evening begin.
No, it really is incredible. I mean, it's just such a wealth of data, and so important for us to really understand what's happening with people in the district with living with HIV. Amanda, you've done such a great job. I wonder if you have suggestions for how I see a couple of maybe junior investigators on the call. Are there what are the best ways for people to come up with ideas and come to you? Are there things that you would suggest, or opportunities for them to apply for K grants, for example, or I know people probably apply for pilot awards, but then also taking them to that next level.
Yeah, so most people have just been reaching out, sending us an email that they might have an idea, either to myself or Anne, and then we're able to meet with them and kind of talk through it.
Mhm.
I do encourage people to go to our website because we do post every analysis that we are working on have been published. And so we do try, make sure that they're not duplicative. If they do have a similar idea, then we can work with you to say, well, maybe we've looked at this question, but we haven't looked at this aspect of that particular research question. So that's kind of a good starting place, is to look at what we've done and then to reach out to Anne or I, and we can kind of talk through your ideas and help formulate a research question. You mentioned also K awards. So we haven't had anyone use the data for a K, but it certainly would be exciting if there were someone who had an idea. And we would, obviously be supportive of talking through that with an individual. The current funding for the cohort is an R24, which is a resource grant. And so that's our mission right now, is to provide data to the city to understand how people are doing, but also to provide it as a resource to researchers and to further people's careers. So.
And do you see a way to be able to embed interventions within and then use cohort data as kind of the base for any intervention studies that may be launched?
Yeah, so our current R01 is our test case, where we're doing this cluster randomized trial. We have 13 sites participating, and so we'll be randomizing the sites. But absolutely, that is, this is a platform to be able to do interventions. Our mantra these days is, we're going from an observational cohort to an interventional cohort.
Any questions from anyone else on the call for Amanda? And always, she shared her information, and it will be in the recording. So feel free to reach out to her directly with any questions you may have. But please use this. It's a beautiful resource and something that I think the whole city should be very proud of. It's really nicely put together.
Thank you. And I mean, I think probably half of the people on this call are already somehow involved in the cohort. So hopefully they can, attest to the utility of the study and the data, and they also can answer questions if there are any.
Well, that's wonderful. Thanks so much, Amanda.
Thank you.
So Manya?
Sure. So I'm going to take it away now, and I'm going to introduce you to the DCCTU, this was Alan Greenberg's great brainstorm that we'd have the DCCFAR, we'd have the DC cohort, and then the third would be the DCCTU. So we have a little symmetry there. I'm going to show you just a few orientating slides. Just give me one moment. And then I'm actually delighted that we have some of the leadership of the DCCTU is here with us. So you'll hear directly from them. And Jean Jordan is here, who's the director of the lab, so we can hear a little bit from her if she's willing. I'm putting her on the spot. But this is mostly just to orient you to where we are and what we're doing, because like the cohort, the DCCTU is a terrific resource, and we have 7 years to build this into an even better resource to support HIV-related science here in the District of Columbia. So I'm just going to basically introduce everybody, give you a brief background of the network, because as many of you are experts within this structure, not everybody really knows the history of the networks or how they are structured, and then talk about what we're doing and some of our future plans. And I'm going to go very quickly because I want to ensure we have enough time to introduce to you our research initiative at the end. So I'm going to race through these, but hopefully we'll have time for questions. To give you a little feel for it, for those of you who are not aware of the networks, the NIH-funded HIV trials networks began very early on in the epidemic as a way to consolidate resources and identify sites that could conduct high-quality clinical trials across the nation and eventually globally. And critically, these not only lend support structurally, but they also allow accrual of sample size to facilitate answering the important research questions of HIV. And I won't go into it deeply here, but you can see that it's starting in 1991 all the way to this current cycle, 2021 to 2027, there's been slightly different structures, really in response to the changing epidemic itself. And I want to highlight here, I have a circle here just because I began my own career as a data manager long, long ago in the 92 at the pediatric ACTG. And here we are down here all the way in the 2021 cycle, and we're working on prevention and treatment. But all to say that these structures are quite durable, and I find myself marveling at how different they seem like they should be, but in fact, the structures are almost identical as they were in the 90s. So it's rather remarkable how durable these are. The DAIDS clinical trials networks are not just the clinical trial sites, as you might see in pharmaceutical trials or other networks. They're really a whole web of really centralized resources to support the implementation of clinical trials at numerous sites globally. And this slide here just describes some of the different agencies and bodies that are called to tasks. So there's a laboratory central, there's clinical research support services, study monitoring, the sites themselves, and then data management centers and so forth. And I think it's interesting to notice this because in terms of mentoring junior faculty and even students, for graduate students, it's really important to know that there's tons of great job opportunities, and many people don't even know that these exist, but that the HIV/AIDS Network Coordination Center that manages all of this through DAIDS is quite huge and offers a lot of great resources for us to collaborate with, but also really truly to have inform the next generation of public health and clinical medicine. This current iteration, which was funded just at the end of last year, actually arrived upon a network structure that had a vaccine trials network, a prevention trials network, an AIDS clinical trials group for adult clinical trials, and then a maternal pediatric adolescent clinical trials called IMPACT. And Natella might be able to talk a little bit about that because of course she's an expert in this as well. So it's nice to have all this great expertise on the call. For this particular iteration, there were 35 clinical trials units that were funded. Now, they're globally listed and you can look online to see them. And each CTU, and this is the important part in terms of our structure, each CTU embraces or parents clinical research sites. And each clinical trials unit has between 2 and 8 clinical trials sites. Those sites then in turn affiliate with different networks. So they can affiliate, for example, with one of the 4 networks if they're selected to do so by these funded groups. And it's really truly a testament of this whole group, including the Hopkins group from which the ACTG CRS came, and Sarah is here and she can tell us a little bit about that. The work with Dr. Borden, Dr. Khan, Dr. Benator at the VA, it's really the testament of all this past work since for the GW side, since the 2008 introduction of our clinical trials here at GW, and since 2013, the site over in Whitman Walker. But it's really that these clinical trials activities have been grounded on numerous activities here. And before that, even an incredible site at Georgetown under Prince Kumar. So I think it's really important to notice that this is really an extension of the foundational work that's done by this whole community over this entire time. I also just want to highlight, because many people don't fully understand how these networks were leveraged. The idea behind the network structure is that they'd be leveraged so that they could facilitate research on a turning on a dime. And of course, when COVID happened, everybody did do that. And the COVID Prevention Network and the active group for treatment were established to allow the DAIDS-funded network sites to participate, among other networks, in treatment and prevention trials. So it's important to notice that these sites, our own CTU and our own clinical research sites, are now not only HIV treatment, cure, and prevention, but we're also COVID treatment and COVID prevention. So before I introduce you to the real stars of this show, I just want to show you a little bit about our aims, which are to provide scientific leadership and administrative infrastructure, to implement best practices to ensure quality conduct of our clinical trials, to engage with our local community, And then finally, and that's I think why we're here today with the CPS Corps, is to capitalize on these resources from the DC cohort, from the DC CFAR, and really start engaging all of you on the call, start mentoring early stage investigators to engage with the clinical trials infrastructure, and start introducing people to this really wealth of scientific inquiry that many times people don't even know exists. So we're interested in engaging more and more people into the clinical trials practices here in treatment and prevention, with the idea that that will help really train the next generation to be involved with clinical trials. Now, with that, I will stop, and I would like to turn it over. I'll stop screen sharing to, I'll introduce you and then you guys can talk. We're very excited to have Dr. Gary Simon, Dr. Sarah Henn. The three of us are the CTU leaders. Dr. Mark Siegel is the HPTN CRS leader, so he leads the research site, and Sarah leads the treatment site at ACTG. Dr. Jean Jordan is here, and she is in charge of the lab operations for all things DCCTU for both sites. So maybe I'll turn it over to Gary first, then Sarah, then Mark, then Jean. And you can say a few words about your current research and your current thoughts about the structure and what it can offer this group.
Hi, everyone. I'm Gary Simon. We have been an HPTN site under the direction of Mark Silver for a while, and we have been running a lot of treatment studies for HIV, usually through pharmaceutical companies. We continue to do both, but the marriage, so to speak, with Whitman Walker and under Mania's leadership, we're going to move into doing some more of the ACTG type studies. Right now, we are literally overwhelmed with COVID activities, such that we finished the Moderna vaccine trial. We're starting the Sanofi vaccine trial. I suspect there will be more vaccine trials, more vaccine administration, let's put it that way. I don't think there's going to be trials with additional vaccine elements unless it's a new vaccine. By that, I mean, I suspect that we will be administering modified Moderna vaccine to some of the participants because of the variants. But we'll have to see how that sorts out. We're looking forward to expanding. Right now, we're restricted by the combination of, not even by funding, combination of space and personnel to run all these things. But I think as things quiet down a little bit, we'll be expanding beyond that. I think this is really a unique opportunity with Mania and with Sarah to really expand the HIV clinical research activity within the city of Washington. I'm really looking forward to it. And beyond that, I'm getting hungry. I bet everybody else is. So I'll turn it over to Sarah.
Yeah, I think I would just echo what Manya and Gary have already said. You know, I think the CTU, the new CTU structure we see as a great opportunity to leverage all the expertise here in DC. You know, I've had the privilege to get to know a lot of you through the CFAR, and this CTU, I hope, will be an extension of that. And it's nice to bring everything more local, and we're looking forward to kind of the collaboration that will allow for us, because you know, even though Baltimore isn't that far away, it's certainly, even 30 miles, it makes collaboration more difficult. So we've been an ACTG site for the last 7 years, so have been focused largely on treatment trials through the ACTG, in addition to some of the industry trials we've done around prevention and treatment. So we're looking forward to expanding that and perhaps getting to do some HPTN work as well. So, and I guess if Mark wants to.
I was going to say, am I next?
You're next.
I'm Mark Siegel. I'm the CRS leader, and I'm very grateful to Manya, Sarah, and Gary for getting the CTU. I feel like since I've been at GW, really the fact that the School of Public Health was asked to be join the HPTN has allowed us to do these really amazing prep trials, prevention trials that really changing the way that we are managing to prevent people from acquiring HIV. And personally, it's been very amazing for my career that I was started as a medical co-lead, went to a PI. I've just been asked to chair one of the upcoming HPTN trials. So it's definitely helped my career development. And I hope that through the CTU, we will bring on other studies that will allow younger investigators to take on kind of the same roles that I've had and be able to develop their careers. And so I think it's invaluable. I also think that being able to work collaboratively with Whitman Walker on some of the ACTG studies will be extremely interesting. I think obviously we have interest in both prevention and treatment. And also we have patients that would be great candidates for a lot of the trials that they're looking at enrolling. So I think we have great resources for enrolling patients, helping them enroll patients, and vice versa. They obviously are a great resource for all things prevention. So I think it is, has boundless opportunities for young investigators here. And I think it's going to be a wonderful thing for GW and Whitman Walker.
Great. Thank you all so much. I think this is really exciting. And Jean, I don't know if I'm totally putting you on the spot. I don't know if you're able to talk right now. Oh, she's on.
You're on
Yes.
your bike.
She's
I
on.
was about to say, Jean, you were going to be on your bike.
You are right, but I am here and I'm totally happy to talk.
Thank you, Jean. Just tell us a little bit about your lab. It'd be great.
Yeah, no, thank you so much. So my laboratory is got a split personality in that we have many wonderful collaborations with 3 of the 4 HIV clinical trial networks, some of which are at Whitman Walker Health, or now Whitman Walker Institute, I guess, and others here at GW. So we do a lot of sample processing for all of these HIV clinical trial networks. We also have been involved recently in the Moderna trial, also a few other COVID-related projects. In addition to sort of supporting many collaborators, we have worked with Amanda Castell on the molecular epi project doing HIV sequencing. And that was a really fun project to kind of get started from the ground up. My R01 is on human papillomavirus with the focus on the MSM population living with HIV, and we're looking to try to identify biomarkers that could help sort of predict high-grade lesions in anal cancer, such that we can help to prevent unnecessary, sorry, I'm outside, prevent unnecessary high resolution endoscopy and biopsy procedures. So that's kind of the goal. I have the privilege of working with Dr. Stephen Abbott on that project from Whitman Walker, and I just absolutely love his team. So those are the main things. And I'm going to be working with Mimi Gosh on another HPV related grant in the a population of trans men. So that will be a really interesting project that will be starting soon. So thank you for the opportunity to share all of that.
Thank you, Jean, and thank you for being so flexible and showing us the outside and your bike. That was awesome. That added a little spice to it. But it's really great you're here because you're one of the selling points of the CTU is that the ACTG CRS and the HPTN CRS already worked collaboratively with Jean, who's provided DAIDS approved processing and more functions than ever could have happened without Jean. So because of that, we got all of our ability to have economies of scale. And I think that was a real strength in our application. So I think that it's really great that we can continue this work together.
We love working with you. Thank you.
Love working with you too. And as I hope all of you can see, I know many of you, and I know I won't even call everybody out because I've worked with every single one of you practically, and I look forward to finding ways of collaborating. And the whole purpose of the CTU really is to engage further. And I think now that we have this great resource and you know the people, hopefully it'll be easier to come approach us if you have ideas for collaboration, if you want some training, if you want to learn about the clinical trials infrastructure, if you want to try to chair a study or be a part of a study team. there's a lot of opportunities for engagement. So we can't spend too much more time, but because we want to take you to the last lap, which will be telling you a little bit about the CPS Core Initiative. There will be a further tool for people to get additional resources for their research. But before we go on, I want to see if there's any questions for any of us about the CTU.
Manya, I have a question. So uh, is there the possibility of someone who's not connected to this CTU and the networks getting access to actual data?
So one thing that is so great, and actually it's yes, current data, not usually, but there's a wonderful public access process, and that's something that Sarah, Mark, Gary, and I can help facilitate. The ACTG has a huge, just like WISE, just like the DC cohort, a huge data request process. So there's tons of ACTG studies. Actually, we're not part of the impact group, but I think that those impact or pediatric ACTG data requests are still possible. The HPTN has a huge public access. So anybody who wants those are free, really well documented. And usually they take a, like a concept sheet type of approach, but sometimes even some of the early HPTN studies don't even require that. So that's a long way of saying yes.
Great. And it sounds like it's not necessarily that complicated or difficult.
No, and it depends. Sarah, do you use that at your site? Do people access those data? I'm assuming it's not for current data because the HPTN doesn't allow that, but the ACTG has a huge repository of data. I don't know if you guys use that.
Yeah, we haven't accessed it. So I would, love to dig into that with someone if they had an idea and work through what that process looks like.
Actually, this would be a great initiative because we can even write grants on using those data sets. And that'd be nice for the CTU we can support junior faculty to do that. So yeah, absolutely. I think even MTN, the Microbicide Trials Network, was not refunded. All those trials moved to other of the groups of the network coordinating centers, but the MTN also has data available. Okay. Did anybody want to add anything else, or should we move on to the research initiative? Okay.
Sounds
Well,
good.
if you have questions, please just let us know, and we will be happy to work with you and your teams to connect you with the CTU. Okay, so I'm gonna just begin here, and then Larry and Sebly and Sam will jump in. These are some of the ways we can engage, but we just discussed those. So this is the idea that we want to present to you. It's a new CPS Core Initiative. And the purpose of the C²M² is that it will help CFAR investigators with your research. And we want to describe how that will happen and then provide a little bit of an explanation of it. And we're excited to share this with you. I'm having a little, oh, great. Thanks, Sarah. That's great. Okay. So the idea behind our new initiative is to match CFAR investigators with skilled trainees and research support. Now, we've been actually doing this in various ways. Sam, most of all, has been doing this. He matches biostatistics practicum consulting students all the time. I've been doing this for years where I'll send an MPH student to help somebody, but it really hasn't been formalized. So what we're doing here now is we're going beyond sort of passive introductions to sort of active sort of matchmaking. And that's our job with this initiative. So through this match, we're going to support the development of preliminary data, grant proposals, and we're going to help you and your teams contribute to the peer-reviewed literature. This is going to be something, and I'm going to show you how it's going to work in a second, that's going to be best for projects without tight timelines, and also things that are not highly complicated, and also things that you don't have paid resources for. So this can fill a gap in your existing research portfolios or for your team members or colleagues. So to give you a little bit of examples, currently, one of our MPH students is doing her practicum with Tony Young and the CEG team. And she's doing this amazing, we had this idea to do a systematic review of diversity of samples in basic science cure specimens. So she's almost done. I got the draft of this paper today. She did this with Tony and Tony's team. And this has been a wonderful match. We're providing Mac Scholar assistance. So Dr. Coy, who is working with Nutella, we are going to be matching her, and Sam has already set up someone, I understand, to work with her to help through this type of process. We also have numerous COVID-19 people in place in the MFA, all around departments of health, all over the DMV, working to do analysis. And these are all graduate students primarily, and we've matched them. So they're helping expand existing teams. And this has been really, really productive so far, but we're hoping to take it to the next level by really structuring it. So the way it's going to work, there are two paths. One is people can get a form on the CFAR website, which is not there yet, but will be there. It'll look something like this. They will send it to Larry, who's our core manager. She'll identify with us a suitable junior faculty or students, and then the connection will be made. I have a form, which I'll show you, we can email later, which tells you all the types of things that can happen. For example, it might not work at first, might not be a good fit. It's kind of like a blind date. So our job is going to be to make sure it's a good fit and to help ensure deliverables are met and so on. The second path is a little bit less formal. We might identify, we all in the CPS core, we do numerous consults with people. And so we might identify this as a way you could sort of expand your service needs. And if we do that, then we'll basically link you to the core manager directly, and then go from there without the form. So there's really two ways of doing it. So I'm going to stop there and let us talk about this and see if there's questions. And Sam and Sebla and Larry can chime in about their thoughts on this and how this will work.
So, so let me just emphasize that this goes beyond data analysis and data management, and so just helping with a literature review. Manya, do you have other? I think you had a couple of other.
Yeah, literature review, data entry, data management. We could help with, also with meta-analysis has happened. It doesn't have to be just analytic. That's exactly right.
Another good example is we had someone from Howard actually who was very interested. She doesn't work in HIV, but she does a lot of work on mucosal immunity, and she's more like a biophysicist, and so she works on actual mucus and mucus characteristics. So for her, it was matching her with a clinical or research cohort. So it's matching someone with a good idea to help them find populations that they can work with. If they're in the basic science world, it's linking them to the clinical, to the correct, well-defined phenotyped people who can contribute samples. So it's really linking people across from access to samples, access to data, access to analytics, but then also trying to help people get to a finished product that will allow them to build their career and go to the next level. So we're actually really excited about this. Again, organically, it is what we have been doing, but now we're just trying to systematize it a little bit, put some structure to it. And I think we'll be able to help a lot of people with this and hopefully increase even further the productivity. So I know a lot of you are seniors, so please be prepared for us to send people your way. And then if you have needs for someone who can support projects you're working on, if you let us know, then we can also be on the lookout when we, all of us are teaching master's level and PhD level students, postdocs. And so we'd love to be able to provide these bridges to people and be intermediaries. So.
Any questions?
So Mania or Sebla or Sam, I mean, do you see any potential of expanding also this into whether or not individuals that are not so involved with the basic science, but they need some either understanding some new assay or trying to find something that there is a potential for helping from the basic science core or even the basic science investigators. It doesn't have to be the core, but it can be, something like that, that people that maybe are not that even in the part of the clinicians where they are not maybe so involved in assay development or assay or running an assay for research purposes. So,
Absolutely. Absolutely. I mean,
Yeah.
so for example, with one of our junior investigators, Blair Spence, she's a great clinician, but we're having to link her in with basic scientists. So when she wants to run beta amyloid and tau, we're linking her into labs to be able to do that. But again, Jean, yourself, either assistance to actually get, if there's funding for you to be able to do run those assays, for training people. Some people may be really interested to get into a wet lab and learn, maybe not right now when I know everyone is a little restricted in how many people you can have around. But yes, in time, it would be to help people develop some of those skills that makes them be able to market themselves better for their next project, right? So to really have an understanding of what does it take to actually do an HIV sequence, for example, and to learn some of the analytics, even though they don't end up in time. I think having some basic understanding of those things makes them that much better suited. So I think it will expand and have a lot of tentacles into different areas, and maybe something that, you're at the Basic Science Corps may want to build something like this to be able to field students into your labs, because I know a lot of sweat comes from trainees to actually do some of this work that we do. Yeah.
I think the more we can expand this model, it helps everyone. And there's a lot of people, I mean, and I'm sure it's true for basic scientists as well as people like clinical and behavioral epidemiologist types like me. There are probably lots of people who actually really need help, but they don't have a huge grant, but they have a good idea and it would help them get a grant. And so I'm sure that's true across the board in all the CFAR cores. So I think that we would definitely want to expand this model to whatever it is people need. So absolutely.
Sylvia here. I just think that this is a fantastic thing, and I can see it as sort of all-encompassing across the cores. Really very good. I hope you're writing it up on your annual report for the CFAR. But also when we get together with our quarterly meeting of the cores and the developmental core, I think that this is something that would definitely go all over, and I think it's great. I'm glad I attended. Thank you.
Yeah, spread the word, because you're in the perfect spot to let people know if you're mentoring them. So let them know. We want to be, and for community organizations too, this is a great, I mean, we've done this over the years informally, but this was the first time we've done it more formally with Tony. And, people need evaluators, people need data entry people, they need outreach people, and there's a lot of ways this could be leveraged. So spread the word. We think it's great.
Well, actually, also, it might be a way of people not understanding that they need something as well, right? Just knowing that it's there.
That's
Very
right.
good. Great.
Exactly. Thanks, Sylvia. That's great. Well, great. Well, I don't know if there are other questions. No? Well, we're going to post this all online, and I'm sure maybe Patty and Lorena can help us get this in the news, the weekly update at one point. So you'll get all this information, but please keep this in mind and let us know if you have questions in the future or just want to talk with one on us, and we can talk with you to help work it through.
Thank you very much.
And with that, I guess I'll turn over to Subway to take us on home.
Yeah. So thank you so much. It's just so nice to see everyone, to have these discussions. And, I think this is just, it's planting a seed, but I think what we'll try to do is maybe let you know of what we have planned for our next few meetings, so you are aware. So, for the ones where you really want to attend, and then also send people that you think would benefit from being a part and hearing some of these talks and discussions. And then as you see, we'll send you, I think we have things outlined for about 6 months. But then if there are things where you want to get space either on a specific day or even beyond that 6 months, we can build this calendar out more. And if you find that, things that are useful, please let us know. Things that you think we can improve on, we always would like to hear about that. We're not stuck and set on doing this way. This is something that can evolve and change. And we want it to be very inclusive. And again, we want it to allow people to build collaborations and use what we have, but also expand what we have and increase the productivity of everybody, but especially our young and junior investigators, and kind of keep bringing people in to fill in. So thank you so much. And I think we can maybe end a little bit early if nobody has any other comments or questions. Oh, we got kids with us. That's awesome.
Hi. Well, great. Well, thanks to everybody. Thanks for joining.
Yeah, thank you so much, guys.
Thank you.
Have a good
Thanks,
evening.
everyone.
Great to see
Bye.
you.
Everyone.
Bye.
It was great to see
Bye.
everyone.
Bye.
Bye.
Thanks for
Nice to
hopping
see
on,
you,
Jean.
Jean.
That was so nice.
Good night,
Super.
everybody.
Bye. Thanks, everybody.
Bye, everyone.
Thanks. Bye.
Bye.
Bye. Thanks, Mark. Thanks, Larry.
Oh, thank you. Can I had a request for your
Oh, sure.
slides. Can I,
Oh, sure.
I
Can
can,
let me send you the new ones because I think I sent it. I'll send you what I think is the most updated batch.
okay.
Yeah,
Okay.
yeah. Thank you so much. Did you feel okay about it all?
Oh, yes, I did. I wanted to make sure I didn't miss anybody coming in later. So I see that, but it's fine because the green admit button comes up even when someone else is sharing the screen. So
I learned that. I was always so worried about, because now that we've been teaching by Zoom, I was always so worried that I'd be lecturing and couldn't see it, but it does come up like you can't really miss it. So it's
Okay.
good. Yeah.
yeah, because
I was worried about that. Wow. Well, this is great. So I'll send you the slides, and then we can get that to Lorraine, and we could send the form out, and they could work their magic on sending it to the weekly update.
Yes, great. And then too, maybe we could talk later about the match program, about how I can best, be of service to you and identifying students and whatnot. Okay.
Probably, I'll be interested if we get a lot of requests. I think that's going to be kind of exciting. I bet it won't happen, but it could. But I think at the beginning, probably what we'll do is, if you get a request, maybe just send it to you, me, Sam, and Seble, and the four of us can just try to talk about how to approach it.
Okay.
So you're not going to be in this yourself. It's not like you're going to have to, because I think we're going to have to, we can know where to point people to. So it won't be all on your shoulders by any stretch.
Well, great. It sounds really exciting. And I would have loved to have had something like that as a graduate student, an opportunity like that. It's amazing. Yeah.
I think it gives them such great experiences. Like, I know that the ones we've given these types of matching to on the informal over the years, they get the best jobs because they have real life experience.
Yeah.
And they get a publication or a poster, or they just get some, something great on their CV that they really did that was meaningful. Plus, it really does help the investigators. Like, if you needed somebody to help you on your STAR project, you could be like, okay, I want a research match, and we get you somebody. We send you our pictures.
Oh, okay.
Right.
Well, thank you.
Yeah, it's great to see you. Have a great day. Thanks again for organizing all this.
Okay. You're welcome.
Bye. Have a good night.
Bye-bye.